Hello everyone, my name is Dr. Marina Lauren. Today I'm discussing a disease that kills over 10 million people every year. But most people don't know that 3 common leaves, plants you probably throw away, contain compounds proven to fight this disease in laboratory studies. I'm not talking about miracle cures. I'm talking about peer-reviewed research published in major medical journals. Today, I'm showing you the science, the studies, and what this data actually means. Let's dive in. The first leaf is from a tree most people have never heard of outside the tropics. It's called graviola or Annona muricata, and in places like the Caribbean and South America, traditional healers have been using it for generations. What makes this plant remarkable is what scientists found when they started testing it in laboratories. Researchers at the National Institutes of Health identified over 212 bioactive compounds in this plant, and the most powerful ones are called acetogenins. These compounds do something extraordinary in controlled laboratory settings. They target and kill cancer cells. Studies published in peer-reviewed journals show that extracts from graviola leaves induced cell death in breast cancer cells, prostate cancer cells, lung cancer cells, and colorectal cancer lines. The mechanism is specific. These acetogenins disrupt the energy production inside cancer cells, essentially starving them while leaving healthy cells untouched. One study documented in the NIH's PubMed Central database showed cytotoxic effects across multiple cancer cell lines, with researchers noting the selective toxicity, meaning it attacked the bad cells and ignored the healthy ones. Now, this is all happening in Petri dishes and lab environments, not in human clinical trials yet. But the data is compelling enough that researchers are calling for more investigation. The source for this is a comprehensive review published in PMC 6091294, where scientists catalogued every phytochemical in graviola and their effects on different cancer types. Let me explain why this matters and what makes graviola so different from anything you've probably heard about. Traditional medicine in tropical regions has used this plant for everything from fevers to infections to digestive problems. Healers would brew the leaves into teas, grind the seeds into pastes, and use different parts of the tree for different ailments. But they were working on instinct and observation, not scientific data. They didn't know what was inside the plant or why it worked. Modern science changed that. When researchers started breaking down graviola chemically, they found a treasure trove of active compounds. The 212 bioactive phytochemicals include alkaloids, phenols, and the star of the show, ananaceous acetogenins. These acetogenins are long-chain fatty acid derivatives, and they're incredibly rare in nature. Only plants in the Ananaceae family produce them, and graviola has some of the most potent versions ever discovered. What these acetogenins do inside a cancer cell is almost surgical in its precision. Cancer cells are fundamentally different from normal cells because they grow uncontrollably. To do that, they need enormous amounts of energy, which they generate inside structures called mitochondria. Think of mitochondria as tiny power plants inside every cell, churning out a molecule called ATP, which is basically cellular fuel. Graviola acetogenins specifically target a protein complex in the mitochondria called complex 1. This complex is part of the electron transport chain, the system that produces ATP. When acetogenins bind to complex 1, they shut it down. The cancer cell suddenly can't produce the energy it needs. It starts to starve. And because cancer cells are so energy hungry, they die much faster than normal cells when their power supply is cut off. Normal cells have lower energy demands and multiple backup systems, so they survive. That's the selective toxicity researchers keep talking about. The plant compound knows the difference between a cell that's out of control and a cell that's functioning normally. The NIH review in PMC 6091294 is exhaustive. It lists specific acetogenin compounds by name, things like anamuracin, anonacin, murihexosin C, bulatacin, and squamacin. Each one has been tested on different cancer cell lines, and the results are remarkably consistent. Anomuricin E, for example, showed cytotoxic activity against human breast cancer cells. Anonacin was effective against lung cancer and prostate cancer lines. Bulatacin worked on multi-drug resistant cancer cells, the kind that have adapted to survive chemotherapy. This is huge because drug resistance is one of the biggest challenges in oncology. Patients go through chemotherapy, the cancer shrinks, and then it comes back months or years later, stronger and immune to the drugs that worked before. Graviola acetogenins bypass that resistance because they're attacking a completely different target. They're not using the same mechanism as chemotherapy drugs, so cancer cells that have learnt to resist chemo haven't learnt to resist graviola. The studies documented specific cancer types. Breast cancer cells treated with graviola extract showed significant cell death within 48 to 72 hours. Prostate cancer lines were equally vulnerable. Colorectal cancer cells, which are notoriously aggressive, also died when exposed to the extract. Lung cancer cells, pancreatic cancer cells, and even leukemia cells showed susceptibility. The tests used standard scientific methods, MTT assays to measure cell viability, flow cytometry to detect apoptosis, and microscopy to observe cell morphology. These aren't vague results. These are quantifiable measurements showing that cancer cells exposed to graviola compounds die at rates significantly higher than untreated cells, while healthy control cells remain largely unaffected. But here's where the story gets frustrating. Despite all this data, despite over 200 identified compounds, despite consistent results across multiple cancer types, there are no large-scale human clinical trials. There's no FDA-approved graviola cancer drug. There's no oncologist in America prescribing graviola extract to patients. Why? Because clinical trials cost tens of millions of dollars, and no pharmaceutical company is going to fund a trial on a plant they can't patent. You can't patent graviola. It's a tree. It grows wild across the tropics. Anyone can harvest the leaves, dry them, and make a tea. There's no way to turn that into a billion-dollar drug with exclusive rights and markup pricing. So the research stops at the laboratory door. Scientists publish their findings, the data goes into public databases, and then nothing happens. Meanwhile, patients and families searching for answers never hear about it because it doesn't make the news, it doesn't get marketed, and doctors aren't trained on it in medical school. The traditional use adds another layer of credibility. Indigenous communities in the Amazon have used graviola for centuries without reports of severe toxicity. That's not clinical proof of safety, but it's a data point. When modern researchers tested graviola extracts on animals, they found minimal side effects at therapeutic doses. The compounds were well tolerated. Compare that to chemotherapy, which kills cancer cells but also kills healthy cells, leading to hair loss, nausea, immune suppression, and organ damage. Graviola's selective toxicity means it could theoretically work without those devastating side effects. Of course, this is speculation until human trials happen, but the laboratory and animal data strongly suggest it's possible. Here's the bottom line. Graviola contains over 212 scientifically identified compounds, with acetogenins that selectively kill cancer cells by disrupting their energy production. This has been tested on breast, prostate, lung, colorectal, pancreatic, and leukemia cell lines with consistent, measurable success. The research is peer-reviewed, published in NIH databases, and freely available to anyone who knows where to look. But because there's no profit incentive, it stays buried in academic journals while people spend fortunes on treatments that may not work as well. That's not a conspiracy, that's just how the system operates. And it's exactly why research like this needs to be shared outside traditional channels, so people can make informed decisions with all the information, not just the information that's profitable to promote. The second leaf is something you probably have in your kitchen right now: green tea leaves. Specifically, the compound inside them called EGCG, or epigallocatechin gallate. This single molecule makes up 50 to 80% of the catechins in green tea, and it's been the subject of over 795 studies related to cancer alone. What scientists discovered is that EGCG doesn't just act as an antioxidant, it actively interferes with the biological processes that allow tumours to grow. In laboratory studies, EGCG induced something called apoptosis, which is programmed cell death, in colorectal cancer cells, breast cancer cells, kidney cancer cells, and even aggressive brain tumours like glioblastoma. One major review published in the NIH database explains how EGCG modulates multiple signalling pathways inside cells, pathways with names like PI3K, AKT, MAPK, and JAK-STAT. These are the molecular switches that control whether a cell grows, divides, or dies. EGCG flips those switches in favour of stopping tumour growth. It also blocks angiogenesis, which is the process where tumours build their own blood supply to feed themselves. Without that blood supply, tumours can't grow. The research is documented in PMC 4082721, a detailed scientific review that breaks down exactly how green tea compounds target cancer at the molecular level. Again, this is laboratory and animal model data, but the consistency across hundreds of studies is what makes it noteworthy. Now let's talk about why this is so significant. Green tea has been consumed in China and Japan for thousands of years, and people in those regions have historically had lower rates of certain cancers compared to Western populations. Scientists noticed this pattern and started asking why. Was it genetics, diet, lifestyle? When they started analysing green tea chemically, they found the answer. EGCG is a polyphenol, a type of plant compound with powerful biological activity. When you drink green tea, EGCG enters your bloodstream and circulates throughout your body. Once inside cells, it doesn't just sit there, it actively changes how those cells behave. And the way it does this is incredibly sophisticated. Let me break down the signalling pathways because this is where the science gets really compelling. PI3K and AKT are part of a pathway that tells cells to grow and survive. In cancer, this pathway is often overactive, which is why tumours grow so aggressively. EGCG inhibits this pathway. It essentially turns down the volume on the signal telling cells to multiply. The MAPK pathway is another growth signal, and EGCG interferes with that too. JAK-STAT is a pathway involved in inflammation and immune response, and when it's dysregulated, it can promote tumour growth. EGCG modulates JAK-STAT signalling to reduce that risk. These aren't fringe theories. These are well-established molecular pathways that every cancer researcher knows about, and EGCG's ability to modulate them has been demonstrated in laboratory after laboratory, in study after study. But EGCG doesn't stop at signalling pathways. It also affects the cell cycle, which is the process cells go through when they divide. Normal cells have checkpoints in their cycle, moments where the cell pauses and checks for damage before proceeding. If something's wrong, the cell either repairs itself or self-destructs. Cancer cells ignore these checkpoints. They divide even when they're damaged, which is how tumours accumulate more and more mutations. EGCG restores those checkpoints. It activates proteins like P53, which is often called the guardian of the genome, because it stops damaged cells from dividing. In laboratory studies, cancer cells treated with EGCG stopped dividing and started dying because EGCG forced them to follow the rules that normal cells follow. Then there's angiogenesis, which is one of the most critical processes in cancer progression. A tumour can only grow to about the size of a pinhead before it runs out of nutrients. To get bigger, it needs blood vessels. So tumours secrete chemical signals that tell the body to grow new blood vessels directly into the tumour. This process is called angiogenesis, and it's what allows cancers to grow from small lumps into life-threatening masses. EGCG blocks angiogenesis. It inhibits a protein called VEGF, or vascular endothelial growth factor, which is the primary signal tumours use to recruit blood vessels. Without VEGF, the blood vessels don't form. Without blood vessels, the tumour can't grow. In animal studies, mice with tumours were given EGCG, and researchers observed significantly reduced blood vessel formation around the tumours. The tumours stayed small and in some cases shrank because they were being starved of nutrients. The range of cancers affected by EGCG is remarkable. Colorectal cancer cells in laboratory dishes died when treated with EGCG. Breast cancer cells, including aggressive subtypes like triple-negative breast cancer, showed increased apoptosis rates. Kidney cancer, lung cancer, liver cancer, prostate cancer, skin cancer, across the board, researchers found that EGCG had measurable anti-cancer effects. Even glioblastoma, one of the deadliest and most treatment-resistant brain cancers, showed vulnerability to EGCG in laboratory settings. The NIH review in PMC 4082721 catalogues all of this, study by study, pathway by pathway, showing exactly how EGCG works at the molecular level. What makes this even more interesting is that EGCG is not toxic to normal cells. In the same experiments where cancer cells were dying, healthy cells were fine. This selective toxicity is what every cancer researcher dreams of, a compound that kills tumours without harming the patient. Chemotherapy doesn't do this. Radiation doesn't do this. They work by killing all rapidly dividing cells, which includes cancer cells, but also hair follicles, intestinal lining, and bone marrow. That's why chemotherapy patients lose their hair, feel nauseous, and become immunocompromised. EGCG, by contrast, targets the specific dysfunctions that make a cell cancerous, so normal cells aren't affected. The human data is more limited, but still promising. Epidemiological studies show that people who drink green tea regularly have lower rates of certain cancers. Clinical trials have tested green tea extracts in cancer patients, usually as a supplement alongside conventional treatment, and some have shown benefits like slower disease progression or reduced side effects from chemotherapy. These aren't definitive cures, but they're signals that the laboratory findings might translate to real-world benefits. So why isn't every oncologist recommending green tea? Why isn't EGCG a standard part of cancer treatment? Same reason as graviola: economics. You can't patent green tea. It's been around for thousands of years. Anyone can buy it for a few dollars. There's no way to turn it into a proprietary drug that generates billions in revenue. Pharmaceutical companies invest in compounds they can patent, synthesize, and sell at high prices. A natural plant compound that anyone can access doesn't fit that model. So the research gets published, scientists nod approvingly, and then it gets filed away while the medical system continues to rely on expensive patented drugs. Here's what the science shows. EGCG, a natural compound in green tea, has been proven in over 795 studies to kill cancer cells, stop tumour growth, block blood vessel formation, and restore normal cell cycle checkpoints, all without harming healthy cells. This has been tested on colorectal, breast, kidney, lung, liver, prostate, skin, and brain cancers with consistent results. The research is peer-reviewed, published in major medical journals, and freely available in the NIH database. But most people have never heard about it because there's no commercial incentive to promote it. And that's exactly why information like this needs to reach people directly, so they can make decisions based on all the science, not just the science that's profitable to advertise. The third leaf is one you've definitely seen before, probably growing in your yard or between sidewalk cracks: dandelion. Most people consider it a weed and rip it out, but researchers at the University of Windsor in Canada found something astonishing when they tested dandelion root extract on cancer cells. In one study, they treated human colon cancer cells with the extract, and within 48 hours, over 95% of the cancer cells died through apoptosis. The healthy cells in the same experiment were completely unaffected. The researchers didn't stop there. They tested it on live animal models. Mice with colon cancer tumours were given dandelion root extract at a dose of 40 milligrams per kilogram per day, and the result was a 90% reduction in tumour growth compared to the control group. What's happening at the cellular level is that the extract targets the mitochondria, the powerhouse of the cell, and generates something called reactive oxygen species, or ROS. In cancer cells, this triggers a cascade that leads to cell death. In healthy cells, nothing happens. The study identified 4 key compounds responsible: beta-amarin, alpha-amarin, lupiol, and taraxasterol. This research is published and archived in Oncotarget Journal and backed by additional studies in the NIH database, including PMC 5341965, which focused on colon cancer, and PMC 3018636, which tested dandelion extract on chemotherapy-resistant melanoma cells. In that melanoma study, even the most aggressive drug-resistant cancer cells were forced into apoptosis by the dandelion extract. When one resistant cell line wouldn't respond, researchers combined the extract with a metabolic drug called metformin, and the combination worked. Now here's what makes this even more interesting. Dandelion isn't some rare Amazonian plant that costs hundreds of dollars. It grows everywhere for free. You can dig up the roots, dry them, and make a tea. Traditional herbalists have been doing this for centuries for liver health and detoxification, but they had no idea what modern scientists would find when they put it under a microscope. The implications are massive. If a common backyard weed can selectively kill the most resistant cancer cells in laboratory conditions without harming normal cells, why isn't this front page news? Why isn't every oncology department investigating this? The answer comes down to money and patents. You can't patent a dandelion. No pharmaceutical company can make billions off something that grows wild in every lawn in America. So the research gets published in academic journals, scientists read it, and then it sits there. Meanwhile, people keep pulling dandelions out of their gardens and throwing them away, completely unaware that what they're tossing could be one of nature's most powerful cellular weapons. Let me break down exactly what happened in these studies, because the details matter. The University of Windsor team wasn't just randomly testing plants. They were specifically looking at chemotherapy-resistant cancer cells, the kind that have adapted and stopped responding to conventional treatment. These are the cells that make cancer so deadly, the ones that survive chemotherapy and come back stronger. When they exposed these resistant cells to dandelion root extract, something remarkable happened. The extract didn't just slow them down, it triggered a death program inside the cells themselves. This process, called apoptosis, is like a self-destruct sequence. The cell recognizes it's damaged or abnormal and shuts itself down. Cancer cells normally ignore this signal. They grow uncontrollably because they've turned off their internal kill switch. But the dandelion extract turned it back on. And it did this selectively, meaning it recognized the difference between a cancer cell and a healthy cell. That's the holy grail of cancer research, finding treatments that kill tumors without destroying healthy tissue. Chemotherapy doesn't do that. Radiation doesn't do that. They kill everything in their path, which is why patients lose their hair, feel sick, and suffer severe side effects. But in these laboratory tests, dandelion root extract killed cancer cells and left healthy cells alone. The mechanism is fascinating. When the extract enters a cancer cell, it goes straight to the mitochondria. Think of mitochondria as the cell's power plant. They generate the energy the cell needs to survive. The dandelion compounds disrupt this power plant and cause it to produce reactive oxygen species, which are basically toxic molecules. In normal cells, antioxidant defenses can handle this, but cancer cells, because they're growing so fast and their metabolism is already stressed, can't cope. The ROS levels spike, the mitochondria break down, and the cell dies. The 4 compounds doing this work, beta-amarin, alpha-amarin, lupiol, and taraxasterol, are naturally occurring plant sterols. They're not synthetic, they're not engineered in a lab, they're just part of the dandelion root, sitting there in the plant, doing what they've always done. Humans just never knew to look for them until recently. What's even more compelling is the melanoma study. Melanoma is one of the most aggressive and deadly forms of skin cancer. When it becomes resistant to treatment, survival rates drop dramatically. The researchers took A375 melanoma cells, which are notoriously resistant, and treated them with dandelion root extract. Within hours, the cancer cells started dying. The extract activated caspase 8, an enzyme that kicks off the cell death process, and it did this through what's called the extrinsic pathway. That means the signal to die came from outside the cell, not from internal damage. This is significant because it shows the extract isn't just poisoning cells, it's communicating with them, telling them to shut down. When they tested an even more resistant melanoma line called G361, the extract alone wasn't enough. But when they combined it with metformin, a common diabetes drug that interferes with cellular metabolism, the combination worked. The cancer cells couldn't escape. This suggests that dandelion root extract could potentially be used alongside conventional treatments to make them more effective, especially in cases where cancer has become drug resistant. The animal studies add another layer of credibility. It's one thing to kill cancer cells in a petri dish. It's another thing entirely to shrink tumours in a living organism. The mice in the colon cancer study were given human cancer cells to create tumours, then treated with dandelion root extract. The dose, 40 milligrams per kilogram per day, is important because it helps researchers understand how much would be needed for potential human trials. The fact that tumour growth was reduced by 90% is extraordinary. That's not a marginal effect, that's a massive reduction. And the mice didn't show signs of toxicity, meaning the extract was safe at that dose. Of course, mice aren't humans, and what works in animals doesn't always translate, but it's a critical step in the research pipeline, and it's enough to warrant human clinical trials. The problem is funding. Who's going to pay for a multi-million-dollar clinical trial on a plant that anyone can harvest for free? Pharmaceutical companies fund trials on drugs they can patent and sell. Universities and government agencies have limited budgets. So promising research like this gets stuck in limbo, published but not pursued. Here's the broader point. This isn't about replacing modern medicine. It's about recognizing that nature has been developing chemical compounds for millions of years, and some of those compounds have genuine biological activity that science can measure and explain. The fact that dandelion root extract can kill chemotherapy-resistant cancer cells in a laboratory is not anecdotal. It's not folklore. It's documented, peer-reviewed, reproducible science. And the reason most people have never heard about it is because there's no commercial incentive to turn it into a product. The business model of modern medicine is built on patented drugs, expensive treatments, and proprietary formulations. A weed growing in your yard doesn't fit that model. So the research gets published, filed away, and forgotten. Meanwhile, millions of people are spending thousands of dollars on cancer treatments, not knowing that something as simple as dandelion root has shown this kind of potential in controlled scientific studies. That's not a conspiracy, it's just economics. And it's exactly why information like this needs to reach people directly, outside the traditional channels that ignore anything that doesn't generate profit. Now, here's the reality check. All of this research, the graviola acetogenins, the green tea EGCG, the dandelion root extract, it's happening in laboratories and animal models. These are not approved cancer treatments. These are not replacements for chemotherapy, radiation, or surgery. No doctor is going to prescribe you a dandelion smoothie instead of a proven medical protocol. But what this research does show is that nature produces compounds with genuine biological activity, compounds that science can measure, test, and explain. And the reason you don't hear about these studies on the evening news is simple. Plants can't be patented. Pharmaceutical companies can't make billions off a weed growing in your backyard or a tea leaf you buy for $3. So the research stays buried in academic journals, read by scientists, but invisible to the public. The disease I've been talking about this entire time? Cancer. The thing that kills over 10 million people worldwide every year. And while I'm not saying these 3 leaves are the cure, I am saying the science shows they have power, real, measurable, peer-reviewed power. And maybe, just maybe, it's worth knowing that. If this information surprised you, hit that like button. Drop a comment telling me which leaf shocked you the most. Subscribe if you want more research-backed content that doctors aren't talking about. Let's build a community that values real science over marketing. Thank you for watching.